Hey everyone, today's lesson is going to be over domain, archaea, and bacteria. So our learning objective for this one is we're going to compare and contrast the characteristics of the domain, bacteria, and archaea. Now, like I said, there are a lot of similarities between these two, and that's why we're going to discuss a little bit of the differences and why they are separated. So archaea and bacteria, these two domains are made up of prokaryotes, but there are some differences in structures and functions that distinguishes them from one another. In a previous lesson, I told you that archaea were actually classified as bacteria up until pretty recently. And now we've found that archaea, in fact, if you look at this phylogenetic tree, archaea are actually more closely related to humans, animals, fungi, basically anything that's a eukaryotic cell. They're actually more closely related to that than they are to bacteria. So we'll look into that a little bit further about why that's the specific case for archaea. So, archaea, these organisms are ones that usually inhabit extreme environments. So, this is the number one thing that separated out archaea from bacteria, and one of the number one things you'll hear people talk about that explains the difference between them. So, archaea live in extreme environments like deep sea vents, saline waters, which is just super salty, hot springs, and even below petroleum deposits. So, these are places where not a whole lot of life can actually exist because there are such extreme temperatures or concentrations of solutes and things like that. But these organisms have figured out how to survive within those places. So archaea, they do lack a nucleus like bacteria because they are prokaryotic cells, but they have metabolic pathways and enzymes more related to eukaryotes. So these metabolic pathways that I'm talking about are things like cellular respiration and also protein synthesis, these enzymes that are acting more closely related to eukaryotes. So that's why on that phylogenetic tree, they were a little bit closer to humans than they were to bacteria. And then we also know that they reproduce asexually. So that means archaea, even though they do have a lot of these same metabolic pathways, they do not undergo mitosis or meiosis. So this is one of the ways in which they are very similar to bacteria. They're going to undergo a division process called binary fission, which is very, very similar to bacteria. So, bacteria, these are the smallest living entities in the world and they're microscopic. You cannot see bacteria, all right? Bacteria can also, a lot of people just call them germs because these are the things that can cause disease within us. And this is what people are actually referring to is bacteria that are around us in everywhere that we go. So these are only observed under a microscope by performing a number of staining techniques. So this is different things that scientists do in order to see them better. So these are all pictures of them underneath the microscope, especially this one right here. You can tell you would not be able to really see these bacteria unless we used a staining technique. So we just put a dye on the slide itself in order for us to be able to see the organism a little bit better. So some bacteria are beneficial and help in various purposes like antibiotic production, industrial use, and biogeochemical cycles. However, we do know that some are pathogenic and cause mild to severe diseases. So bacteria are not necessarily always good or always bad. It can depend upon the bacteria itself. We know that certain strains of E. coli can cause people to get sick, but there are bacteria that live within your gut that help break down food and absorb more nutrients for you every day. So they can be good or bad depending upon what specific strain and bacteria it is. And we also know that almost all bacteria have a cell wall made up of peptidoglycan that protects them from harmful chemicals. So this peptidoglycan is what's surrounding their cell membrane and is giving them some extra strength. And that also is how we kind of distinguish different bacteria themselves. Okay, you can see here there's a couple of different shapes that bacteria have. So this first one over here is what we call spirella. It is a spiral-like bacteria. These ones are long and cylindrical, and then you have spheres. So bacteria come in a number of different shapes, sizes, and this peptidoglycan layer can also change, and that's how we normally are going to be distinguishing all of these bacteria from one another. So that's it for this lesson, and go explore further.